is my supervisor is Lily. She's also the head of the laboratory. And in today's talk, I will show you my work entitled From Freshwater and Marine Habitats to the Laboratory, Exploring Kleptoplastic Dinoflagellates. So what does kleptoplasty mean? So to answer quickly this question, I show a little schematic I did about Nostodynium aeruginosa, which is a good example, a good model for kleptoplasty. So kleptoplastic dinoflagellates such as Nostodynium is able to feed on a photosynthetic prey. Here it's Chromonas, and it is able to keep the plastid from this prey, and we call this the kleptoplast, and it will keep it for several days up to several months, depending on the species. And why do we study the kleptoplasty? It's because we think it's a good model to study plastid endosymbiosis, which is a process by which an eukaryotic cell engulfed and permanently retain a photosynthetic symbiont that we call the plastid now. So we think that kleptoplasty is kind of like the intermediate stage before the plastid become like permanent. And doesn't work. Okay. So kleptoplastic dinoflagellate are really hard to cultivate, and only few cultures are available. So it will be like kind of big challenge during the PhD. And it's also hard to confirm the kleptoplasty, especially because we work from environmental samples. So we cannot be 100% sure that the organism we're focusing on is kleptoplastidic. So during the page, I would like to answer two main questions. The first one is what is the hidden diversity of kleptoplastic taxa, and what is the diversity of kleptoplast integration? So I will divide, as I said, my presentation in like freshwater habitat and marine habitat, and I will first talk about freshwater habitat because our lab is located in Czech Republic, in České Budějovice, which is like 1 hour 30 from Prague. And apparently, Nostodynium has been found in some lakes around Czech Republic, and this, I think Nostodynium is also able, you can also find it in like so many lakes all around the world. So with my colleague, we went sampling from October to January in some lakes around České Budějovice, and we actually found Nostodynium in one of the lake, which is called Velký Zvolenov. My pronunciation is probably really bad. Sorry. So as I said, we went sampling from October to January. So the temperature of the water was quite different, like from 0 to 17 degrees, but we were still able to find Nostodynium, which was quite impressive and nice because usually we go sampling during spring or summer. And we collected the fraction above 20 micrometer, and this is where we found Nostodynium. Now that we found Nostodynium, now the goal was to try to establish a culture of it. So we knew, thanks to the literature, that this organism is able to feed on the Chromona species, for example, coming from this culture collection. So we order it. And in parallel, I did a single cell isolation of Nostodynium, thanks to the inverted microscope. So I isolated, I don't know how many cells to try to have a culture. So in each well equal to one cell. So it was kind of like the protocol. So now I encountered some challenges, of course. So the first one is that I couldn't really have a culture of Nostodynium at the beginning. So I was using AF6 medium, but at the beginning in my recipe, I was using tris buffer, which is not the original one. Usually we use MES, but in the lab at that time, we only got tris, and it really never worked. So I switched to MES buffer, which is the original recipe, and it did work. Like I got this kind of culture. So the small green dot are the prey, so the chromonas, and the big cells swimming are the dinoflagellates, Nostodynium. So now that I have this in my well, the goal was to scale up. So when I was scaling up to a 6-well plate, it usually don't work because I think it's a bit stressful for the cell. Like the difference of the volume is maybe too big. So I was switching to 96 to 24-well plate, and actually it did work. So now that I have this culture, the goal was to try to maintain it, and maintain in plates. It's not really convenient, like you have to inoculate every 2 days, like almost every day, because the prey overgrow super quickly. So I wanted to use some flask, so I used tubes and 50 ml flask. The tubes, it's really the best one because Nostodium stay really in the bottom of the tube, and the like the prey uh, stay like swim everywhere. So it was really convenient to inoculate because I was just taking a pipette and just taking from the bottom of the tube and transfer in another tube. Whereas it was really complicated with the flask because in the flask the Nostodium stay everywhere, swim everywhere, and the prey overgrow super quickly. So it's really hard to inoculate because Nostodium is everywhere. So at the end, the cell was super stressed and I was losing it. But there is one solution to this problem if you really want to stick on the flask, is to use like a filtration setup. So I was using a 5 micrometer mesh that I was putting in the filtration setup, like in the middle of the bottle. And I was autoclaving everything to have a sterile, sorry, I need to drink. To have a sterile environment. Then I was taking my cultures and pouring in the filtration setup, and only the prey was going through the mesh, and the dinoflagellate was staying above the filter. So I was just keeping them and resuspending them in a fresh medium. So now that I have a culture which is stable, I had two main questions at the beginning. The first one is like really wanted to have like a proof that Nustodinium feeds on the chromonas that we ordered from the culture collection, and we were also wondering what does it feed on its natural environment, like in České Budějovice in the lake. So what I did is I had my culture, so I was isolating from the culture I got, and I was also going sampling again in the same lake, and I was directly isolating from the field samples from the lake, and I was doing also the single cell isolation. I was taking a picture of each cell, then transfer each cell in one tube to do ITS 16S single cell sequencing. So the ITS is to do to sequence the DNA of the dinoflagellate. The 16S is to sequence the DNA of the plastid. So what we got is a tree, like two trees. So here's the first one, so the ITS1. So in red you have the Nustodinium coming from the field, and in blue it's the Nustodinium coming from the culture, and in black it's like the published sequences. Published sequences. So what we can see here is like the sequences the DNA are quite similar, which is not really surprising. But what is interesting is like when we also study the 16S tree. So to get this tree is we got the sequence from the 16S of the Nustodinium, the plastid of Nustodinium, the prey, so the plastid of the prey, and also all the rest is also what is available on published papers. And what we can see here, I will do a zoom so you can see better. For example, here in blue, you have with the arrow the 16S sequence of the chromonas coming from the culture collection, and above it you have the sequence from the plastid coming from the Nustodium I got in culture. So we can see that both sequences are similar, meaning that Nustodium feeds on chromonas, like the one we ordered. And when, like to answer the second question, like what's going on in the natural environment, we can see that, so we have to focus on the red ones. the Noodin from the environment, they are able to feed on different species of Chromonas because they come from different subclade for A and for B, and you can see that the sequences is not really similar. So as I said, it means that this cell is able to feed on different species of Chromonas, and they are probably also able to switch their food since they don't eat the same species. So now that we answered the question that Noodin really feeds on the culture, the Chromonas from the culture collection, I really wanted to study more on like the transcriptomic level, what's going on and what are the differences between like the starved condition and the fed condition, especially because in the lab, a lot of people are really expert in transcriptomics, so it's also a good opportunity to do this kind of experiment. And I started with fed culture, so I didn't do the starvation condition yet, but in the future I will do it. So for the fed cultures, I have like several flasks with my cultures. I filter everything with the same process I explained. I keep the fraction above 5 micrometer to only have minus 2 dunium. And in parallel, I was also doing a culture of the prey. So both culture was like around 200 milliliter. Then I did an RNA extraction with the phasemaker protocol, and we sent this to sequencing. We received the results a few days ago, so I will do this, like study the transcriptome in the next weeks. So I cannot say anything else about this, but I just wanted to introduce like the methodology. So now that the transcriptome will be studied, I also would like to focus on the plastid itself and the fate of this plastid. And I think that using expansion microscopy may be a good idea, and it's also something that has not been done yet on Nostudinium. To do so, we have to use like a fed culture with like Nostudinium, of course. We have to fix the cells, and since it's dinoflagellates, it's like really hard to fix them. And I tried several protocols. Like we tried several protocols, and we found out that 3% glutaldehyde is a good one to fix the cells. However, I don't know yet if it will work with expansion microscopy because with through some discussions, I heard that using a high concentration of glutaldehyde, sometimes it may alter the expansion microscopy technique. So I need to find out this. So that's why also in parallel, I also would like to do high pressure freezing because I heard that this one is like the best protocol to do expansion microscopy. So this is what I will try in the future too. So actually, we did a first trial a few months ago, like 2 months ago, something like that, with the help of Magda. So we used at the time not the same fixatives. So those one are not the good one because at the time I didn't know that 3% glutaldehyde was the right one. But still, I think it was kind of interesting to show you this because, for example, so here you have the prey and here you have the dinoflagellate Nostudinium. We can see that first the expansion did work, for example, because like Chromonas usually is like 10 micrometer, like here it's almost 20, and Nostudinium is like 25, and here it's like 50, something like that. So it expanded by 2 roughly. So this is a good point. The second good point is like the NAH ester, like in both organisms, actually it's quite good staining, especially for Nostudinium. We also have the cytox, which is the staining for the DNA, which is also quite good for both. And also, like for example, Nostudinium, we can even see like the chromosomes, and we have a second signal that is maybe the nucleus of the prey. But as I said, I'm not really happy about the shape because like the fixatives are not the best one for this organism. So for the future, what I want to do is I would like to do RNA extraction from a starved culture, as I said. I also would like to do differential gene expression on single daughter cells with the SMART Sec3 protocol. So I will try that. So why I want to do that? It's because actually Nostudinium, when it do when the cell divides. One of the daughter cell keep the nucleus of the prey and the kleptoplast, and the other one only keep the kleptoplast. So it would be interesting to see the difference in the gene expression between both cells. And as I said earlier, I also would like to do expansion microscopy. So now I will switch on marine habitats, so it's kind of different work, and I will explain you what we did. So we went sampling in Villefranche-sur-Mer 1 month ago with my team. We went on a boat to go sampling every day, like every morning for 5 days. Then we were coming back in Villefranche in the lab to do lab work. So what we did is like for some of them, we took some samples and I fixed them with 2.5% butylaldehyde to maybe observe them later on microscopy. But the most important part is this one. We did manual isolation of single cells, so it's like the same process as Nostudium. So in each well, we transfer one cell to one well. So we did this for all the species we found. Then we were taking a picture and then transferring the cell in each tube to do 18S and 16S sequencing. So this one is to identify the host and the plastids of the cells, the dinoflagellates we found. And we also isolated for cultivation and also for single cell transcriptomics. So for this, sometimes we were taking some pictures of the cell we found, and then we were isolating them and transfer them in lysis buffer, then we freeze right away. Or I was doing the cDNA synthesis directly in Villefranche, and then I will continue the protocol here in Czech Republic. So here is like the potential candidate for kleptoplasty. So the first one is Oxytosum constrictum, one of my favorites. I don't know why, but I really like this shape. So this one, there is not many, I mean, almost nothing available on the scientific literature. So here you have, like, for example, a drawing coming from the 19th century. So it's the first person who described this organism. It's Franz Schutt. But there is not many, there is no data available on this organism, so it's quite interesting. And as I said, we did the 16S sequencing to understand where the plastid come from. So in the black arrow, you see all the oxytosome sequences, we got from Villefranche. And we can see that the sequences look quite similar to the aptophyte Phyllocystis cordata, meaning that Phyllocystis cordata is probably the prey of Oxytosum constrictum. Now we have another candidate, which is Podolempas bipes, I don't know how to pronounce it. So here is some like light micrographs I found in the literature. So those cells are coming from the Mexican waters. And here, when we sequence the 16S, so the black arrows, you also see like the Podolempas 16S sequences, and it's also quite similar to to Phyllocystis cordata, meaning that Phyllocystis cordata is also probably the prey of Podolempas bipes, which is quite nice because we can use this organism to feed on different dinoflagellates. Now we have Palaeolophalachroma unicinctum. So this is also the cells we found in Villefranche. It was quite nice because the color of the cells were, sometimes they were super green, sometimes a bit less green. So it's also like a good signal to see if this candidate is potentially kleptoplastidic. And in the literature, we have no paper available yet on this organism, but we know that thanks to my colleague Kazuya, who sequenced this organism a few years ago in Japan, that he found out that the known endosymbionт is Florenschella. And here you have like some light micrographs and fluorescence image of Palaeolophalachroma. And when we sequenced the 16S here, we found out that Palaerophalachroma may be able to feed on straminopile prey, because the sequence from Palaerophalachroma is quite similar to Dictyophyceae, coming from, for example, this one is like it's available on the culture collection of Roscoff, and also this one, Frontella pavula, it's also quite close to this one. So it's also maybe both of them are the praise of Palaerophalachroma, but it's something that we still have to investigate to be sure. And the last one is Phalachroma rapa that we also found in Villefranche. So what was kind of nice is that, for example, you can really see through the microscope that the color of the plastid is different between both cells. I was also wondering if we can see that through the trees, and we will find out. And thanks to the literature, we were able to see that, yeah, it's really Phalachroma rapa. So I just put like a paper of where the images, like the scanning electron microscopy images are coming from. And when, like this organism, as I said, is a bit not like the others because it feeds on apparently two different preys. One is coming from aptophytes, so you can see like B3, this cell is probably like the sequence of the plastid is quite close from Chrysochromulina, so it was probably this plastid. Cannot be 100% sure, but it's a good sign. And for example, this cell, which has like brownish, more like brownish color, actually this one we know that it's coming from a straminopile symbiont, but we don't really know which one. That's the thing. So I think we need to investigate a bit more to have this answer. So what we can see here, like the final conclusion is like Phalachroma rapa feeds on several species, and they're coming from two different phyla. So it's this species is this Phalachroma is probably not specialist, but more like generalist. It can feed on different species. So for the future, I would like to identify the endosymbiont of the observed dinoflagellate by 16S. So have to repeat everything to also check if we get the same results, if we discover something else. I would like to get a culture for each potential kleptoplastic candidate. For example, Podolampas bipes with like phoscistis, like trying to feed Podolampas with phoscistis and see if it works. So to do so, I need to go sampling again. And I will also continue doing single-cell transcriptomics from the isolated cells. So I would like to thank you for having me in this DinoSphere seminar, and I also would like to thank my colleagues, sorry, who helped me a lot during this year. Thank you.
Thank you, Leïla. That was very cool. Yeah, okay, we've already got a question from Richard.
Hi, really, really nice work, Leïla. I'm sorry if I missed part of this. It's a public holiday here in France. I'm making vegan stuffed peppers at the same time, but what I followed was really, really, really exciting. My question for you is whether you plan to use co-occurrence data at any point to see whether, particularly, for example, Cytidinium is always found in the same place where Chromonas is found. This might also be quite useful to differentiate specialist versus generalist feeders in your data. And my second question, because I always have two, is for your 16S trees, have you considered for your marine data using the metagenome assembled genomes found in Tara oceans? Because there's a lot of plastid genomes that have been assembled from wild data, and you might be able to start to see some of these plastids as really belonging to dinoflagellate endosymbionts.
Okay. First question. So for Nostudinium, actually, Chromonas, we found it like everywhere in so many lakes. Like even I remember I used to go sampling and found Chromonas. I was like so happy. I was like, okay, I will find Nostudinium, and actually I couldn't. So even in some lakes where there's the food, we cannot find Nostodymium. So it was really in this specific lake that we found it. And for example, I went sampling again a few weeks ago to the same lake, and I still found Chromonas, but I couldn't find Nostodymium anymore.
Okay.
So
They may not overlap with one another. I guess really the question would be kind of more the inverse direction. If you have a specialist feeder, it will only ever be found in sites where its prey is found, even if the prey can live without the predator. So it might be interesting to see if there are certain taxes that are always found in the places where you do see Myzozoan.
yeah, true. Um, um, and now I have to answer the second question, right?
Well, you don't have to, but
Yeah.
I was just thinking about plastid mags in Tara, whether it'd be worth relating them to your 16S trees.
Yeah, I think I have like we did this tree like few days ago, and I think we can investigate more to get like data from like I did not found the data from Tara. I think it's a good idea, and I think I need to do that. I need to put like many more data on those trees because I know that so many things are missing. But yeah, this is something that I will do for sure.
If you want to talk to somebody about this, I'd recommend you contact Malas Jami in Sweden. I think she'd be delighted to help you.
Mawash?
Jami, Jamy.
Liz, Mawash was at the micro-C course, or it's her?
Yeah,
Ah,
100%.
okay. Okay, I will contact her. Thank you.
Thank you, Richard. Looks like Melissa also has a question.
Yeah, I do. This is really awesome. And I have many questions. So I know you don't have all of them in culture yet, but I'm curious to know for the ones you do have in culture, after the feeding, how long does the stolen plastid persist within the host? And specifically, I'm wondering about rounds of cell division and if it persists within successive rounds of cell division.
Um, so I try to do like starvation experiment, like experiment. I mean, I was just like putting the cell and let them starve. Like I was not feeding them. So I was observing every 2 days under microscope, and I could see the cell. I think the maximum was like 20 days. 25 days, but on the papers it can be even more. But the Nostudinium I have in culture, I could only like see during 25 days. And the thing is like the more the time pass, the smaller the plastid is. So at the end, like after like 25 days, you almost have like colorless cells in the culture, and they are really smaller. My head is like this. It's really like smaller, uh, and are super also fragile, I guess. Like they can die super quickly. So I don't know if it answers your question.
I guess what I'm kind of wondering is that if the host manages to kind of when it divides, if it the daughter cells each receive some stolen plastid or not.
Yeah, each daughter cell receives a plastid, but one of them is like the plastid is bigger than the other. And my theory is like the daughter cell that has also the nucleus of the prey has the bigger kleptoplast. But that's just a theory. I need to like have a scientific proof. But just like me observing on the microscope, I think it's this. But yes, this is what I found.
Really cool. Thanks.
So if there's nobody else who has a question, I'm curious. So this is kind of subjective, but are you just based on your observations looking at stuff in the water or looking at the literature or whatever, does it seem like there's probably a lot more cases of kleptoplastic dinoflagellates out there that we just like don't know are doing kleptoplasty, or are we mostly already targeting the major groups that are doing it? There's no wrong answer. But
The thing is, like, for example, Oxytocin constrictum, like, nobody said that it's kleptoplastidic, I guess. Like, we only have, like, some papers, but just they describe it, like, the morphological shape of the cell and saying, yeah, it's Oxytocin constrictum, but there is nothing on the plastid, like, nothing. So I guess it's, like, maybe a new find regarding the kleptoplastidic field. And I guess, like, I think I need to go sampling again to observe that. But, for example, freshwater, I don't think there are a lot more kleptoplastic dinoflagellate, to be honest. But in the marine waters, I guess, yeah, I guess there are a lot. But it's just like, as I said, it's kind of hard to know, like, this organism is like, on the microscope, like, okay, this one is kleptoplastidic. Like, it's super hard to know that. But
yeah.
but I guess, yeah, I think, like, for example, Oxytocin, there are lots of species of Oxytocin, and a lot of them, when I was observing under microscope, like, they were like, the color of the plastid was a bit different, or sometimes they were more green than the others. So it's also for me like a good sign of kleptoplastidy. But for example, when I came back in the lab and we tried with Kazuya to do like some sequencing of some of them, for example, Scolopax, it never worked for whatever reason. So like some like thing that block us, but still I think we, like, with future sampling campaigns, we will be able maybe to answer this question. But for me, yeah, I think there is diversity. Like, we don't know yet.
Cool. Okay. Are there any more questions for Leïla? Okay. So I think we will move on to our next speaker, Noodin Jacko. Noodin is a grad student reaching the end of their time in the Keeling lab here at UBC. And their research is focused a lot on parasites, dinoflagellates in particular, and other myzozoans that are parasitic to animals and also other protists. So we are going to get to hear about Noodin's research. Noodin, if you want to share your screen. Perfect. That looks great.
Okay, great. Thank you for the introduction, Liz. Yes, my name's Noodin. I'm in Dr. Patrick Keeling's lab at UBC, and I'm excited to share with you my talk today titled A Phylogenetic Investigation into the Plastid Evolutionary Parallelisms within Myzozoan Parasites. So this is a short adaptation of my exit seminar. So I hope I don't go over. But first, I want to acknowledge that a lot of this work that from my PhD was carried out on the ancestral, unceded, and stolen territories of the First Nations people, Musqueam, Squamish, and Tsleil-Waututh nations. And so when you think parasites, you may think complex life cycles infecting humans, like the development of disease. And one such parasite that does that is Plasmodium. And Plasmodium is an apicomplexan that causes malaria in humans. But parasites are much more diverse beyond their human hosts and actually infect a wide diversity of animals in the ocean. And so that's where a lot of my interest lies and what we're going to talk about today. So quickly, just showing you the eukaryotic tree of life. And I want to highlight some lineages that actively have parasites with plastids. So starting off with land plants, we have microheterotrophs that depend on fungal connections for their nutrients as they have significantly lost capacity within their plastids. And then we also, within the archaeplastids, we have rhodophytes. And an example of this is vertebrata that infects another algae in the ocean. And then we also have chlorophytes, and chlorophytes such as this really exciting and fun parasite. I call it exciting fun because I love this parasite. It's called a cladogonium, and it infects freshwater shrimp. It's detrimental to the freshwater shrimp and is a really large issue for aquaculture. And a lot of pet stores get this infection very frequently and end up decimating whole populations. So, oops. But today we're going to focus more on alveolate parasites, specifically dinoflagellates and apicomplexans. These two lineages make up myzozoans, and so apicomplexans are a large lineage of animal infecting parasites. Many are obligate parasites, and then also with dinoflagellates here. This is an example of a parasite called plastidinium that we'll talk about soon. But dinoflagellates are a large lineage of flagellates that are freshwater and marine. As you know, we're in DinoSphere, so everyone is here for the dinoflagellates. But many of them are mixotrophic. However, there's a good section of parasitic dinoflagellates, and that's where a lot of my interest lies. And then I also just want to tie back that earlier my reference to Plasmodium are apicomplexan, so it really is a large lineage of parasites that are quite important. So when we look at oceans, with increasing amounts of environmental DNA sequencing becoming more affordable, and the access to the data is much more prevalent, we have a greater understanding of the diversity and the distribution of parasites in global oceans. So this is actually an image here of or like it is a map of the world with all the Terra Oceans sample sites from this study in 2015 by De Vargas. And when you look at all of those samples and search for their symbionts, if I bring your attention to this bottom bar graphs, when you look at these symbionts in orange, those are apicomplexans, and then in green you have dinoflagellates. So they do make up a really large portion of the symbionts within the global oceans and are really important. And yeah, environmental DNA is really highlighting their prevalence. I also want to bring your attention to that many of these dinoflagellates symbionts in these samples are plastidinium. Okay. So the parallelisms in apicomplexan and dinoflagellates. My first talk is going to be about corallicolids, a cnidarian infecting parasite that highlights evolutionary retention of chlorophyll biosynthesis in apicomplexans. And then secondarily, I will discuss blastidinium, an uncultivated lineage of parasite with parallel losses of photosynthesis. So cnidarians are these wonderful, colorful, vast organisms. This is almost like an ad for my supervisor's Instagram page, but they do harbor many symbionts, specifically and most notably Symbiodiniaceae. However, today we're going to talk about their other symbiont, which are corallicolids. And corallicolids were initially like the project really started when Walden was able to identify the location and the location of corallicolids within their host. So here you can see in red are all of the corallicolids within their host here, and then the first image of corallicolids beyond environmental or amplicon studies. Also, one of the really exciting things that he discovered about corallicolids were that he sequenced the plastid genome and he found that this obligate intracellular parasite still retains chlorophyll biosynthesis genes. So we can see them here. So this was really interesting because, of course, there's no other genes associated with photosynthesis present. So what possibly could these corallicolids be doing with the chlorophyll biosynthesis pathway? So the first question was to really lock down the phylogenomic position of corallicolids, and that was really challenging because the nuclear SSU and the plastid SSU had two different phylogenomic positions. So the plastid SSU in corallicolids were branching early in the apicomplexans, and then the nuclear SSU were branching corallicolids as late in the apicomplexan tree. So what we did was go out and find the best model systems for isolating corallicolids, and in order to essentially get high coverage data and collect transcriptomes so we can do a multi-protein phylogenomic tree. So we decided that Madrasis mirabilis and Parazoanthus were the two optimal hosts. This is because they are 100% infected and they're easy to collect. The benefit of Parazoanthus as a host is that it does not have any symbiodiniaceae symbionts, so it makes it a little bit easier to isolate purely corallicolid cells. So this is what it looks like in the field. We brought all of our microscopes to Curacao, and we collected coral, and I did some cell density gradients to really concentrate the corallicolids from the host cells and from also symbiodiniaceae. And through that, I was able to generate high coverage transcriptomes and confirm that corallicolids branch late in apicomplexans. So they're branching sister to protococcinians here. So now that we've confirmed the phylogenomic position of corallicolids as late, we can map out the parallelisms that have occurred in apicomplexans. So Gregorines and Cryptosporidium have already had 3 independent plastid losses, and we know there's 5 independent losses of photosynthesis here at these nodes. And then because of the phylogenomic position of corallicolids, we're able to identify 10 independent losses of chlorophyll, at least 10 independent losses of chlorophyll. So that was really exciting. And this emphasizes that there must have been a decoupling between chlorophyll biosynthesis and photosynthesis early on in the backbone of the apicomplexan plastid evolution. So do corallicolids retain nuclear encoded plastid target counterparts of the chlorophyll pathway? That was what I went to answer with my new transcriptomes. And what I did was I generated many, many plastid phylogenies with diverse eukaryotic taxa. And I generated many trees similar to what these look like. And through these phylogenies, I was able to confirm that indeed there were nuclear encoded counterparts to these pathways, specifically at least half of them. I find that the identification of at least like the last one is really exciting too, and possibly it's more of getting higher coverage transcriptomes in order to really lock down the whole pathway, is a potential in the future. So why would corallicolids retain chlorophyll biosynthesis? And some people suspect that it has to do with the ecological niche of corallicolids and being on the coral reef, but that would suggest that ancestors of the of corallicolids also all had the same niche. So that's extremely unlikely. It's more likely that there are it's working towards some sort of cellular component of corallicolids. So specifically, a feedback inhibition system that contributes to like regulation of heme, because chlorophyll biosynthesis and heme have share like early components of their pathways. So now I want to bring you to focus on blastidinium. And blastidinium are these really large endoparasites that actively, with many members that actively are photosynthetic within their host. And this is a video of a copepod who's actively alive, and there are two large individuals infecting their host. So I like to think of this as in, imagine having like a rabbit-sized parasite on you at all times, and you just can't escape it. This is unfortunately what many copepods in global oceans endure throughout the year. So the thing that's really interesting about blastidinium is that there are pigmented and non-pigmented members. So it's a really great system to investigate the loss of photosynthesis. So in order to do so, I went out and sampled in like around BC and also in Curacao. I did hand toes, plankton toes. I've looked at deep toes from like 200 meters depth. And usually this is what the plankton looks like. I will essentially just kind of search through the plankton, finding any copepods that are struggling with swimming. That's a really good indication that they have an infection. And I will pick them up with like a Irwin loop and do a micro dissection and collect the individuals and any of the remaining sporocytes that come from the host. Oh. So here are some of the infections that I have taken. I've probably collected over maybe like 60 infections, and also with the help of some colleagues in my lab as well. So it's been a really exciting project, and through these these collections, I was able to generate many transcriptomes and ultimately put them in a multi-protein phylogenomic tree. And with that, I was able to identify the parallel losses of photosynthesis across blastidiniales. So everything that has a colored box behind it are transcriptomes that I collected or that has been collected for this project. And they all branch within the paradineales. So First, I'm just going to bring you through some of the clades. First, PC1 refers to pigmented clade 1, pigmented clade 2, and then non-pigmented clade 1, and this is a mixed clade. So I'll show you some of the pictures so that you can get an idea of what these look like. This first clade here was actually an infection of 15 individuals in a single copepod host. So these happen to be just the highest quality transcriptomes that I could include in this data set. And then we're going to jump down to this mixed clade, which is really yeah, it's a really interesting, bizarre clade where you have cells that are actively pigmented and then sporocytes that have no pigmentation whatsoever. So perhaps it's actively in like a loss of photosynthesis. And then we have this really beautiful clade because these images are so nice and was collected by Liz in Alaska. So this is a non-pigmented clade 2. And the coverage of these transcriptomes in this data set are pretty exceptional, considering, you know, we can't do any culturing, and this is all environmental sampling with and transcriptomes using SmartSeq. So similar to the corallicolid project, I investigated the presence, absence of plastid genes using single gene phylogenies. And in this case, I was investigating the retention of photosynthesis. So these are each like filled in square has been confirmed through a phylogeny and presented here. So the non-pigmented clades like clearly are not expressing any photosynthesis related genes, and that also is including this mixed clade as well. This mixed clade, however, is really interesting because it does retain the PET complex. And so I need to do a little further investigation on this clade in particular. And another really interesting result from collecting so many different Blastidinium from Curacao and in Canada was that this one individual actually through multi-protein phylogenomics and also 18S with replicates is branching outside of essentially where all the Blastidinium are clustered. And we suspect that this Blastidinium indeed is actually not Blastidinium, and rather it evolved the same life strategy as Blastidinium. And I'm going to rename this organism as Yemadinium after the sea goddess Yemaya in Curacao. So it's quite exceptional considering it's also infecting copepod hosts and visually has multiple like cell types, which is quite characteristic of Blastidinium infections. So we have our sporocytes here, and this is like layers of sporocytes and potentially what would be considered the gonomere here, but is challenging to fully say so. So through this project, I was able to uncover 3 parallel losses of photosynthesis in Blastidiniales. So specifically here at the node of non-pigmented clade 1 and both the non-pigmented clade 2 and the mixed clade, as well as the new Yemadinium clade. Through this, we also were able to identify an origin of parasitism. here as well. So through these two parts of my PhD thesis, there has been multiple parallelisms that have occurred in myzozoan parasites through my research. So need to highlight that parallelisms are more common than we think. And through the corallicolids, we were able to see the parallel loss of chlorophyll biosynthesis of up to 10 times that happen in the backbone of apicomplexans, and that ultimately it makes sense that chlorophyll biosynthesis and photosynthesis had been decoupled early on in apicomplexan history. And then with my blastidinium project, there had been at least 3 losses of photosynthesis and a parallel origin of parasitism in blastidinium as well. So I would like to thank all the wonderful people that I got to work with over the years and some people who are in the chat today. So I will leave it there and have time for questions as it is exactly 10 a.m. here in Vancouver.
Thank you, Noodin. That was really cool. I've been waiting to hear like the synthesis of all of this
Me too.
data.
Yeah.
And that was super, super interesting.
Super creepy.
Yes. Also very creepy.
Yes, yeah, the videos.
So glad I'm not a copepod.
Yeah. I like the rabbit size.
Yes. Yeah, I tried to estimate how big it would be for us,
Yeah.
but then you said rabbit, and I was like, okay.
Yeah, maybe even larger because, yeah, they're, huge.
Are they, when there are several in there, like 15, are they all like the same size? Like
They're all the same size, yes. Hmm.
How does this work? And but a copepod is still alive then? I mean, functioning?
It is still moving. Yes, it's still alive. Mind you, I guess like the 15 individuals will be smaller than, say, a morphotype of just like a one individual infection. But mind you, they'll all be like the same size. And I think it does put like a lot of pressure internally on the copepod, and it bumps out quite a bit. So
And when they release the dinospores, they do that into the first break open the copepod, or how does this work? I guess it died, no, when they release the spores.
So from what I've seen, is that there's like a cuticle that surrounds all those different cell types, and the cuticle will break and the sporocytes will like slowly leave from like the anus of the copepod, and then they become biflagellated again. However, I know Noriko has observed like the whole individual like just popping out of the copepod. I guess it depends like how well the copepod is doing, because if it is decaying or dying, perhaps like there's other means in which the individual will be released. But when the copepod is alive, from what I've seen, they just kind of like in a line just kind of come out.
And the copepod survives that?
It's hard to say because it's really hard to keep these copepods alive. They're wild copepods. I've tried to culture them. They only survive for so long, but it's hard to say what happens in the wild if these Blastidinium are able to leave and then they survive. But from the most part, from all the previous studies, they do die.
Yeah, but at least they don't get infected again. Yeah.
Yeah.
Imagine you have to do this twice.
Yeah, that's true.
Wow, yeah, no,
That's crazy.
super. And cool that you found this like other Blastidinium. Yeah.
Yeah.
Do you expect to find more like Blastidinium-like organisms?
Well, I'm gonna search like environmental databases to pull out sequences and put them in the 18S tree with those transcriptomes I have. The nice thing is that from that day I sampled and had the Yamadinium, I have multiple different individuals that all branch in the same place. So the goal is, yes, to find more. Mind you, like it may be challenging, but yes.
Sorry, I'm having talking back questions
Yeah,
here.
no worries. I
Perhaps
also
like
have a
some
question.
more Terra Oceans, downloading 18S from Terra Oceans.
Noted, did you see that? So with it looked like the clade of Blastidinium that you had was pretty like the phylogenomic difference, like the branches were fairly long. It seemed like it was a pretty like distantly related group across. Did you see that the morphological, like I don't know, it's hard to tell from, I mean, you kind of gave samples of the morphologies, but could you see if there was like really noticeable differences in just sort of the morphotype of the different groups, other than the pigmentation, or was it, I guess that would be hard considering they're always like in very varying states of development or whatever, but could you, like if you saw one like a parasite out in the wild, would you be able to look at it and say like, oh, I think that one's from this clade just based on the way it looks?
Um, yes, you can. There definitely are descriptions of this through Elfskovgard and even like really old like Chaton literature. But specifically for this Yamadinium, um, it does look different from the other Blastidium infections I've seen. However, at the time, I didn't realize because I was just collecting as much as I could. I wasn't paying, like, I guess, enough attention. And also, like, now that I look back and look at the photos from what I see, there doesn't seem to be like a clear visual representation of like a trophocyte or even the gonomere. There is like a larger cell which could have been the gonomere and which creates all the sporocytes that kind of wrap around. But the fact that it might just be like the photos too. So it's hard to say. Um, entirely, like if I'll see it again and be like, yes, that is the new one, but perhaps it's one of those pattern recognition things. The more you get and the more sequences confirm its position. Yeah, I see we have a hand up by Richard.
Thank you so much for a really, really thoughtful and considered presentation. And for those keeping track, I have just stuffed my last pepper, so woohoo for that. My question, because I was really taken with your point about parallelism being a recurrent process across biology. And for me, parallelism is kind of like an evolutionary experiment with multiple independent replicates. And I'm going to use that to ask you a very, very specific biochemistry question. Have you looked into mutation events in any of the photosynthesis-associated proteins, either in corallicoids or in blastidinium? Because my suspicion is where you have clades that are repeatedly moving to heterotrophy from photosynthesis, there are things that are going to happen in the sequences of their chlorophyll biosynthesis enzymes that will change their function. And that's very, very interesting to entomologists. So I just wondered whether you'd started to look into the individual protein coding genes to see if there's weird stuff happening in some of these species.
I haven't looked really in depth into the protein coding. However, from the phylogenies, it's really evident that the chlorophyll biosynthesis genes in corallicoids are highly divergent. There's specifically CHLM is really long branching, and it took quite a lot of like thought to really confirm that that one is indeed like CHLM in corallicoids, but I think there is actually potentially a lot to look at there. For my thesis, I didn't dive too deeply into it because I have other projects, but I think it would be a really great thing to look at the protein coding and consider all the evolution that these organisms have gone through and potentially like what other roles that they're playing in the cell biology of cryolacolids.
If you have a free half day and it's a half day question, you could just try putting it into AlphaFold and seeing what the protein looks like.
Okay. Yeah, that sounds fun. I'll do that.
But get topology, go woo, and then nobody knows what it means, but at the very least you can see, does it look, does it fold in the way chlorem normally would, for example? And that can be the ground for somebody, if they're functional genomicists, to then try and test its endometic activity in the distant future. But great job, and thank you for bringing the need to consider diversity and repetition very much to the fore in your presentation. Great job. Thank you.
Thank you.
Thank you, Richard. Does anybody else have any questions for Noodin? Okay, so with that, I guess we will conclude for the summer. Yeah, we will be back again in September. We have not yet determined the exact date, but you will be getting an email of some kind if you are on the mailing list. Hopefully, everybody here is. Please continue to spread the word about DinoSphere and encourage people to tune in and reach out to us if they want to be on the mailing list. And yes, if anybody is interested in signing up to give a talk in the future, I think we are mostly booked through the rest of the calendar year, but starting in 2027, we are going to hope to fill more spots. So feel free to reach out if you want to give a presentation or know somebody who would like to, or if you have nominees.
Exactly, just rope someone in.
Yes, coerce people into giving talks for us. But yes, so unless there's anything else that I'm not thinking of, mostly it.
Yeah, so yeah, you can always check the website. We have the Discord channel, but yeah, otherwise we will contact you by email for the next seminar. And also, yeah, if you have any questions regarding your project, any methods, yeah, please reach out. We try to connect you with someone who might be able to help you.
Yes, the community building aspect of DinoSphere is something we want to keep cultivating. So yeah, so think of it as a little network that you can use. Yeah, and thank you all for joining us, and we will see you in September.
Yeah, have a great summer, everyone.
Stay cool.
Okay. Bye-bye.
Thank you. Bye.
Bye.
Thank you so much.
Bye. Bye, Richard.
Bye.